for a streptacular protein purification. So Strep-tag affinity purification is a way that we can purify proteins by adding a little tag, a little string of amino acids to the end called a Strep-tag that mimics biotin. And then we have this resin, these little beads that streptactin, that basically they mimic streptavidin, which is a biotin binding partner. And the reason why they choose these two is because they have this really, really strong, really specific interaction. And they engineered these though so that they're better for this purpose. And so you can get your protein to specifically bind to the resin while the other proteins are going to flow off. And then you can wash all this other stuff off, and then you can elute or like push your protein out by adding another biotin mimic, disthiobiotin. And disthiobiotin looks like biotin, so it's going to bind to those biotin binding sites in the resin and kick off your protein. What's really cool though is, so biotin is this little like molecule-y thing. We can just add a few amino acids onto the end of our protein and it can still bind to this biotin site. It's pretty cool. So I'll tell you more about how this Strep-tag system was developed, as well as how it works in practice, some differences between different types of resin. So they have also this like XT stuff. I've never tried it, but it binds tighter. It can be good if you don't have a really low yield protein, but the regeneration is a little different. So Strep-tag isn't as like popular as the His tag, but there are a lot of benefits. Better specificity is the main one, and it's good for things where like His tags combine, the His tag resin can bind a lot of things non-specifically, especially for mammalian and insect cells. So I tend to use a His cell when I'm doing bacterial expressions and the Strep-tag when I'm using insect cell expressions. So let me tell you more about how it works.
So say there's a protein that you want to study. You can stick the instructions for making that protein into cells and have the cells make it for you. But you need a way to then be able to purify your protein away from all of the other stuff. And by adding this little sequence of amino acids, this little string of protein letters that specifically bind to the specific resin, to these specific beads, this allows us to kind of like have a handle where we can latch on to the protein of interest, and only the protein of interest, because the other stuff doesn't have that special handle. And so this is a form of affinity chromatography. We're adding just a short string of amino acids. And so how could this help us? So basically, we are adding amino acids because that's all we can add, right? So proteins are made up of amino acids. But there's so much variety in amino acids. There are these 20 different ones, and they have all these different properties that even just having a short string of amino acids is enough to give it specific specificity, so that this column is only going to bind to things that are engineered to have this tag. So with the His tag, you can have some problems because proteins can naturally have a lot of Hisses. But with the Strep-tag, it's more specific because proteins are unlikely to have this specific sequence, this specific string of amino acids, compared to just having a lot of histidine. We'll get more into some of the differences between them in a minute. But the basic idea is that you can use these tags as affinity tags. And so you have this tag on your protein. The protein is going to encounter this resin that has the specific binding partner for that tag. Because only the proteins that are designed to have that tag will be able to bind to the resin, then everything else is going to flow through. You can then flow through a competitor that's going to compete for the binding site on that resin, and now your protein is going to come off, but it's going to come off without all of the other stuff that went in with it. Often there's still some things that are non-specifically bound, but this is going to be less typically when you're using a Strep-tag purification versus when you're using a His tag, because you have that enhanced specificity from this specific sequence. And because this resin has been like genetically engineered, they did a lot of testing to try to optimize the binding relationship between the Strep-tag and this streptactin, and it's very strong because it's based on this really strong interaction between biotin and streptavidin that I'll get into in a minute. These two are both really short. So histidine is only like 6 to 8 histidines in a row. Strep-tag is 8 amino acids, but I'll show you, I use this trin strep-tag. So it has like 2 of these with a little linker in between, but both of them are fairly short. And this means that they're not likely to interfere with your further protein using unless you have some sort of fusion partner. So I often have a fusion partner. So this, I put in this like sumo, this protein that the cells really like making, so I can kind of trick them into making my protein if I stick this fusion partner protein in front of my protein. And then what I'll do is I'll stick a protease recognition site. So like I use TEV, so a TEV recognition site, which is so TEV is a site-specific protease that's basically going to recognize the specific sequence of amino acids I put in, and then it's going to cut that sequence so I can cut the tag off and cut this big old molecule off. But when you just have this tag, one of the benefits of having these small tags is that you don't need to worry about cutting off the tag typically. And the strep tag has been designed too, so that it has this, basically, it has a pretty generic, well-balanced amino acid sequence, so it's not going to mess up like your charge or various interactions. This one's affinity chromatography, where you're using this affinity tag chromatography, is often like used as a first step or like a capture step. So basically, because it's using this really specific thing, you want to do it first before you move on to other forms of chromatography, where you're basing on things like charge and size. So by starting with this first step, you can separate out most of the stuff, but not all of the stuff. And depending on your further uses, you might then want to take it through additional chromatography steps, such as with ion exchange chromatography or size exclusion chromatography, and more about those in other posts. But especially if you cut off that tag, you want to take advantage of the tag while it's there, and you want to take advantage of the fact that it's going to be the most specific and give you the most specific like the biggest differentiator between your protein and other proteins. A note about cutting off those tags. So you can actually cut it off on column. So while the protein is still bound to the beads, or off. You typically have better luck when you elute and then cut off the tag. It's easier for it to like access and everything. So you typically have a better efficiency, but so typically if I'm doing it like on column, I'll do it overnight in the cold room, so things are safe. But you can also do it off column. The benefit of doing it on column is that then your tag is stuck to the column. When you're doing it off the column, then you're going to have to purify out the tag as well as the protease, which you can do in further purification steps. Or you can also flow the cut stuff back through, and that way the tag will be recaptured. When I'm doing it when I do the on column, then what I typically do is I take the stuff that eluted and I run it through just in case there was stuff that fell off, and that way all the tag will be bound. But in these cases, you still have that protease to deal with that you might need to then separate further with your chromatography steps. So let's get more into these tags and the strep tag in particular. So the strep tag is, well, it's just a string of amino acids, but it's a special string of amino acids because it's designed to mimic this interaction between biotin and streptavidin. So streptavidin is this protein that is found in this kind of bacteria, and it binds to biotin really well. And so when I'm talking about really well, I'm talking about really, 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 really well. And so we're talking like femtomolar range. So let me just step back and talk about just a second about KD. So KD is a measure of binding affinity. And I have a lot more on other posts, but the basic idea is that the lower the KD, the stronger the affinity. So if you take two binding partners, so like A and B, and you mix them together, they're going to reach some sort of dynamic equilibrium where the rate of binding is the rate of unbinding. And if you measure the concentration of one of the partners that's required for half of the other partner to be bound, that concentration is called the KD, and we often graph it in logarithm form. So the lower the amount that you need to add to get it to bind, to get half of it to bind, the stronger the affinity. So the lower the KD, the higher the affinity. And so the KD is this overall, you can't see this on rate and the off rate, but the KD is going to take into account the concentrations and these rates. So if you have something that binds really tight, it can be really tight either because it's like binding every time it hits it or because it's staying stuck longer when it sticks. But either way, a low KD is going to be stronger affinity. So basically, biotin has a KD for streptavidin of about 10 to the minus 14 molar. So we're talking in the femtomolar range. And this is like crazy, 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 crazy strong. I cannot even tell you how strong this is. This is like the strongest known non-covalent interaction. So an interaction between molecules, not like within the molecule. So not like the bonds holding the molecules together, but the bonds holding the molecules like to one another. Crazy, crazy, crazy affinity. So this is like too high of affinity for our purposes because you'd never be able to get your protein off the column. The idea with this affinity tag is you want to be able to wash everything off and then wash your protein off. But if your protein is so stuck to the column, you're not going to be able to wash it off. And so this is a problem if you were to use biotin, a biotin on your protein tag. Because you'd have, I mean, like you could get it off probably somehow, but you would have to be totally denaturing all of the proteins, which would not be good. So biotin is this little like molecule, it looks kind of weird like this. Doesn't really look like a string of amino acids, right? But what they did was they took like a random library and they made like a bunch of peptides and then looked to see what bound. And that's how they developed this Strep-tag. They found that it was binding in the same place as the biotin, but it was binding with slightly lower affinity. So they scanned a bunch of peptides and found this one that could serve as a Strep-tag. And then they engineered the streptavidin to form a better interaction with it, a more specific, more stable interaction with it. And so that's how they got the streptactin resin. Then they also, to improve the reason why I use that twin strep tag is to improve the to lower the off rate, so to keep it on the resin longer. They found that it was binding really well, but it was binding really quickly. It had a good on rate, but it was falling off. They had a bad off rate, and so this is why they did this is why you can add this twin strep tag, and this is going to give you the benefit of extra avidity. So with the streptactin, you have 4 binding sites, and so this is going to allow you to have this avidity. And so avidity is basically, affinity would be the strength of binding at one site, and then avidity depends on like the strength is the kind of like extra bonus you get when you have multivalency. So when there are multiple binding sites, this way you can think of it, if a protein is binding one place, if it's binding 2 places, one place falls off, but it's still bound to the other place. Well, now it's kind of still there. And so it's more likely to run back into it. So you get this extra benefit of kind of like having one arm tied there so your other arm can't go as far away. And so it's more likely to hit the thing that it needs to bind to. And so this is avidity. And by adding with this twin strep tag, where you put 2 of these tags on with a little flexible linker in between them so that they can actually flex and move and reach that other site, then you can get to multiple to bind, because this streptavidin and the streptactin, they are homotetramers, so they have 4 units for the same units, and each of those has a biotin binding site. And so it has a site that can bind to the tag. So each of these beads has 4 sites that can bind to the tag. So you're able to get your protein to bind better. This also means that you can wash it really, really, really well. So you can wash it under conditions so you can wash everything generic off with leaving your protein on. In some methods, if you wash it too much, you're going to wash off your protein. But with this, because it's bound so tight, you won't wash off your protein. And so when we're talking tight, we're talking about micromolar. So we can see that this is a lot less tight than the streptavidin, but it is but it is still very good. And If you need even better, then they have this streptactin like XT stuff that you actually need to elute with biotin. We'll get more into the regeneration in a minute. But that has an even stronger binding. I've never actually tried it, but they say it's good for if you have a low abundance protein or whatever, because it binds so tight. Okay. And then when we compete it off, so we're going to need to compete it off. And here we're going to need something that is going to bind to the column in the same place, but push it off. So we wouldn't want to use biotin because that is going to get totally stuck on the column. Although if you're using the XT resin, then you do use biotin, but then you use harsher elution conditions. So I'll go into that in a minute. But typically when we're using this normal streptactin resin, what we use to displace it is the thiobiotin. So if we use biotin with just like the normal streptactin resin, then the biotin is going to get like permanently stuck on there and ruin your resin. But if you use this thiobiotin, then we can compete it off with habitai and stuff. So thiobiotin, it looks like biotin, but the thio part is telling us that it's missing a sulfur, so thiosulfur. So it's missing the sulfur part, and therefore it is not going to, this makes it bind less strongly. So dithiotin is going to bind more tightly than our protein, so it's going to be good at pushing our protein off, but it is going to be more weak than the interaction with the streptavidin. With the biotin. Another note about streptavidin is that it and streptactin, it's super duper sturdy. So it can withstand harsh conditions that'll kill most proteins, but you typically don't want to be using those harsh conditions when you're using your protein. But it's good to know that you can wash it harshly if you need to get off stuff. Another note is that it also looks, streptavidin looks and acts a lot like another protein called avidin. Even though sequence-wise they're only about 30% identical, they both bind to biotin, and avidin even binds a little stronger, but avidin has properties that make it so that you wouldn't want to use it. So it's glycosylated, so it has these sugar chains attached. It's positively charged, so it's cationic. It tends to aggregate, so like clump up. There's a neutral version, neutravidin, which is deglycosylated and tries to get around some of those problems. But although avidin binds the free biotin better than streptavidin is actually better at binding biotin tag things. But cool note about the avidin is that it was discovered in egg whites when the farmers were feeding these egg whites to animals, and they were found they had this like egg white injury. Basically what was happening was that these egg whites were filled with avidin, and this egg whites, and that this avidin was actually binding to and sequestering biotin, potentially to prevent bacteria from having that biotin, but we need the biotin too. So biotin is also called vitamin B7, and it's a helpful cofactor or like helper molecule for some enzymes. Another thing about biotin is that because that biotin interaction is super duper strong, it's so strong that we wouldn't want it for our protein purification, but it's good if we want to basically get our proteins to like permanently stick to something, or if we want to get DNA or RNA to permanently stick to something. If we can biotinylate them, then we can use streptavidin to get a super duper, if we can biotinylate them, then we could use streptavidin to get a super, super strong interaction. This is often used to bind proteins or nucleic acids to chips for various binding studies and various other studies. So you'll see this biotin tag a lot in various purposes like that, but you wouldn't want to use it for this purification purpose because you couldn't push it off. So as I mentioned, there's that other stronger bindings, this like streptactin XT. So this has an even stronger binding. And I should mention too that the strep tag, when I was talking about the micromolar, that's for like if you only had one of those strep tags. When you have a twin strep tag, then you have a better binding affinity. But the real difference between these two is, so the streptactin is going to bind more strongly, but it's going to bind, that means that you're going to need to elute it without, you can't elute it with thiobiotin. So typically I elute with like 2.5 millimolar thiobiotin. But for the XT, you actually elute with biotin, which you wouldn't want to do with this. But then you can elute it you elute it with like NaOH. So typically I'll tell you more about the regeneration, but you can do it with this dye called HABA that is going to bind and push the thing off. I find the HABA kind of annoying because it takes forever to wash it out. What I do is I use like a low concentration like of NaOH, and then I add lots and lots of buffer to make sure that I don't mess up the pH. And then I add water, and so yeah, I add buffer through until it like the pH. I use like a pH stick and make sure that it's okay, and then I add my water for storage. But with the streptactin, the regeneration is going to be different, is just the thing to know. And that when I talk about disthiobiotin, that's talking about when I'm doing this with the normal streptactin resin. Another word, so like with this disthiobiotin, so we typically buy these little vial thingies. They're not vials. What you call those, like plastic containers? I don't know. But this little things of disthiobiotin, and so typically we make this like 50x. So we make like a 250 millimolar, and then we add that to our wash buffer, just like a portion of our wash buffer. So normally what I'll do is I'll make my wash buffer. And then I'll make my elution. I'll take a little bit of that wash buffer and I will add the disthiobiotin. So I'll add like 50-fold less of the disthiobiotin. So if I have like 50 mils that I want to make the elution buffer, then I will add a mil of the disthiobiotin. And I keep this super duper separate so that you don't accidentally add it. One time I accidentally added my elution buffer, and then that's a problem because then you've got your disthiobiotin and things, and now your protein's not going to bind because the competitor is already in the protein, so you would have to like dialyze out the competitor. Big pain. But anyway, so you buy these little containers, and typically what you then do is we just prepare the whole vial at once. We make a solution of it. It comes as like a powder. We make a solution of it. And the important thing is that when you make this, and then we make aliquots and then put them in the freezer. What's important is that the pH of this is you need to make sure that you have a stable pH, you have it at the right pH, and it's not going to be like a crazy pH. So when you dissolve this, what you want to do is we dissolve it in like tris pH 8. And so like 1 molar tris pH 8, and then adjust the pH if you need to make sure that it is it's like a physiological pH, or it's the pH of the buffer that you're going to use. It's not going to mess up your pH is the basic gist. So typically we add like 18.7 mils of tris to these vials, and then that'll make it to the 250 millimolar. And then we check the pH, and then we make the aliquots. And keep them in the -20 freezer until we're ready to use them. So let's talk about how we go about all of this in practice. So we're doing a protein purification. We break open the cells, and now we're going to stick them in the ultracentrifuge to spin out the membrane bits and keep our soluble proteins in this liquid. And now we want to, when this is centrifuging, this is when I typically prepare my column. So I need to pack the column.
Comes as a 50x suspension, so you need to add twice as much as the column volume that you want. So I want, says 6 mil column volume, so I'm going to add 12 mils of the resin. And I'm doing this fresh on this prep, but I'll use the same resin multiple times for many times. But I like to use it if this is like a different protein that I normally purify, and so I like to use the same protein. The same resin with the same protein, and not like switch between proteins with the same resin. So now the column is packing. So the liquid, you can see like the resin is getting concentrated down on the bottom, so the liquid can come through, but the resin can't. So it's going to make a nice bead of resin column that my protein can flow through. And then but first I don't want it to flow through in like this buffer. I'm going to equilibrate the column with my wash buffer, which is the buffer without the disthiobiotin. And then I will add I'll do my washes, I'll add the protein, do my washes, and then add the elution buffer. So I like to prepare my column while I'm preparing my protein. So I like To prepare my column, well, my protein is like centrifuging and stuff. And you also don't want to let your resin dry out. So typically what I'll do is I'll equilibrate the resin, and then I'll actually add it to the tube that I'm going to add my protein to with liquid in it, though. You don't want it to dry out. And then I'll add my protein to it. I'm going to do the binding in the batch mode. If I didn't do the binding in the batch mode, you still do it in your column, but you want to make sure that you have liquid. You don't want the resin to dry out.
After I do the equilibration in the column, then I pour it into the tube. So these conical tubes are really nice for this. And so you see what I do is I let the liquid settle. So typically I then I pour a little bit extra buffer when I'm trying to get this stuff out, like the beads out. So I pour it out and I add a little buffer to wash and make sure I get everything out. But then I add extra buffer and I don't want all that buffer because I want to add my protein and we're going to have this volume issue. So then I just pipette out this extra liquid. So let it settle and then pipette out the liquid. And then it's ready to add my protein too. So I just finished ultracentrifuging. So I lysed these cells this morning. I broke them open and then I spun out all of the membrane gunk. And this is the lysate that has the supernatant that has my protein, and also it also has YFP, which is just a reporter for the baculovirus system. So that's not my protein, and so I should see that the yellow won't stick to the column. And so inside of here is my strep-tagged protein, and now what I'm going to do is I'm going to syringe filter it, so suck it up through a syringe, and then a syringe filter before I put it on my bead to flow more smoothly. And so you could also, there are ways that you can do the strep purification on an Acta, and then you definitely need to filter it. It'll go really, really slow on the Acta, I found. Because the Acta is smart, and so it'll adjust so that the flow is not overloading the column, so it goes really, really slow, and you can have problems with clogging and stuff. So basically I like to do my capture steps in batch, so my first purification step, chromatography steps, in the batch mode, or at least in the gravity flow. So basically with you can do with the gravity flow, you can either do it just pour it into the column and then let it flow through, or as I'll show you, I like to do it in the batch mode where I bind all of this into the resin. So I have my resin in my bottle, and then I'm gonna filter this and add it to that and then let it roll. So this is how you just syringe filter it, and normally I can just press it through okay, but today was extra gunky, so I had to bring in the big gun. So this is a cock tube thingamabob to help push it through. Now that it's finally through, you can see that the resin is kind of stuck on the bottom, so I'm going to shake this a little to mix it up, unpellet the pellet, and get all the resin bind. So now that all the pellet is broken up, I'm going to now put it on the hot dog roller and let it bind. So although that relationship is that affinity is really strong, we also have to deal with things about the kinetics and things about the relative concentrations. And so we have a lot of lysate typically for not a lot of protein. So normally I'll do somewhere around 5 mils of lysate. So if I'm doing 4 to 6 liter prep, I'll do 5 mils of lysate. Today I'm doing an 8 liter of lysate, or 8 liters worth of cells. So not 8 liter total lysate, but so I'm going to do 6 mils. If I'm doing a smaller purification, a smaller scale, then I'll have less resin. But because you have so much more of the lysate than the beads, you can do it in the column, but then you're giving the proteins less chances to actually bind to the resin. But if you do it in this batch mode, where you put it on this, you let it this is our hot dog roller. It was broken for a time and it was really sad. But in this way, you're allowing it to have multiple, lots and lots of chances to find those, run into those beads and bind to them. And so typically I'll do this for, I'll let it roll for like half an hour to an hour to really let it bind. And then I'm going to let it flow through.
Time to go into the cauldron and pour it into the column. I can stop this rolling, and now I'm going to pour it into the column. So it's now flowing. It's flowing okay. I can just let it flow through, and if it's having issues, then I can help it aid it along, but put some pressure. But I'm gonna let it try to do it by itself first. It doesn't pop off.
So at this time, you're collecting your flow through, so the stuff that just comes out. And then what you'll do is you'll wash it.
Start flowing through. I like to like suck out the foam stuff. And now I'm gonna start the washes.
And then the non-specific stuff will come out. So I have like my beaker set up so that I have a beaker for the flow through and a beaker for the wash. And then if I'm purifying multiple things at once, I have my columns labeled and I have my beakers labeled. And then I have the tubes ready to go to collect the elutions. So I'll take all those washes. So typically I'll do about 10 columns volumes worth of the wash. And so this is the buffer without the dicyanobiotin. And I will, so if I have like 5 ml column volume, then I'll do 50 mils of wash. And then I will do the elution buffer. You can also test to make sure. So use a Bradford or spectroscopy, some sort of way to check if protein is still coming off. If protein is still coming off, you need to keep washing because there's still non-specific stuff coming off. Once there's no protein coming off, or once you are just like you overshoot it because you already know how much it's going to take to wash it, then you can add your elution buffer. And now you typically, what I do is I give the elution buffer kind of time to kind of push things off. So what I do is I add 2 column volumes worth of the elution buffer. So if I had 5 mils of resin, I would add 10 mils of the elution buffer. But I let like a half a column volume worth through, and I call this my like E0, my zero elution. This should not really have your protein. This should just be like stuff that was kind of like in there already. And then I stopper the stopcock and let it sit for like 10 minutes. And then I let it flow through and I collect this E1. This is going to be the main bulk typically of where your protein is. Then I start collecting fractions. So like I do 1 or 2 column volumes fractions in order to see where when the protein stops coming off. Or sometimes I'm just impatient or lazy and I already know how much it's going to take. And so typically I often end up just adding like I do the 1 elution, I check it to make sure that there's actually protein in there, because if there was a problem, then there wouldn't be protein in there. As long as there's protein in there, then I kind of just like pour in the rest of the elution buffer I prepared. So typically I do like 50 mils worth of elution and then I collect it. If you were doing like, if you just wanted your protein right away and to work with it as is, then you wouldn't want to get like a ton of volume unless you're just really trying to get all of the protein you want. But because my next step is another purification step where affinity, like a cation exchange, so or an anion exchange, so basically an ion exchange where the protein is going to stick to the column, then it doesn't matter if it's in a lot of volume. It might take the run longer, but now it's in the acta, so it's in the purification machine, and so I don't have to stand in the cold room and do things with it. So I don't need to concentrate it first. It's basically the main idea. Although if you're using like a protease and stuff, you don't want it to be too dilute. And so then at that point, you collect your protein and then it can be on to the next step.
So it's kind of hard to see now because it's still destaining, but you have the ladder, then you have I have the ladder, then I have the lysate, and then I have what came off the strep column. So pretty darn pure. And yeah, so it needs to destain. But then I'm going to cut off the tag because it also has the sumo on it. And then I'm going to take it through further purification to remove the TEV and the sumo and all that stuff.
But you need to then remember that your resin is still, you need to deal with your resin. So the great thing about the Strep-tag resin too is that you can use it over and over. So to regenerate it and remove that dicyanobiotin though, you can add this dye, this habit dye, And basically it's this dye, and it's going to displace the tagged protein. And so your protein is going to flow off and this dye is going to get stuck. What's cool about this dye is that it's like yellowish, but when it binds to the resin, it actually turns red. So you can see as you're adding, it's yellow, and then your resin's turning red. If your resin does not turn red, then your resin is probably kaput. So it's a good way too to test that your resin still has good capacity. It should be a really, really dark red. And if it's not, it's time to use some new resin, although you can use this resin a lot of times though. And then you have to wash it out. And it can take a really long time to wash it out though. Just you can also like store it in the dye as well. So as I mentioned, the other main form of like affinity chromatography you probably hear about is a his tag. When it comes to like these small tags, there's also, you can use the fusion partners. So if you have like GST fusion partner, you can use a binding column that will bind to the GST and that sort of thing. And there are other types of fusion partners, but these two are common ones that are really, really small. And so I tend to use the his tag when I'm doing bacterial expression and the strep tag when I'm doing insect cell expression. So with the his tag, what you basically, it's going to bind based on chelating. So basically, if you have a lot of histidines in a row, they can bind to a metal, they can bind to nickel. So if you have a column that's filled with these little resin that's bound to this thing that binds to nickel, now your protein is going to bind to the nickel on the column. There are problems with incompatibility when you're talking about the his tag, in addition to the fact like that you could have lower specificity because lots of proteins have histidines naturally, so you'll have more non-specific binding. And when you're doing all of the watching, because the interaction is not as strong as the interaction with the strep tag, you risk washing off your protein as well. But it's another thing is that with the his tags, you need to be careful. There's some buffer incompatibilities as well as protein incompatibility. So you don't want to use a his tag if your protein binds metal too, because then your protein is going to bind to the column and cause problems. I had that problem once. Yeah, but anyway, and you don't want to use you don't want your buffer to have like DTT, it's going to turn your column brown. You also don't want your buffer to have EDTA, so that's a metal chelator. So basically it's going to bind to and steal this metal. And then if you steals the metal, then your protein can't have it. So those are a couple of reasons why you might want to use one other versus the other.